Find odd with respect to intermediate. Yes. If you learn, that is C is O, K, yes, few, moxa. Check it. M for malic acid, that is this is Krebs cycle intermediate. S for succinyl coenzyme, a Krebs cycle intermediate. Citric acid is Krebs cycle first product. Then where is pyruvic acid? So pyruvic acid is odd with respect to acids of Krebs cycle. These are the sequence of acids in Krebs cycle. You see pyruvic acid is not there, so it is not the intermediate of Krebs cycle. Then enzymes of citric acid cycle already mentioned, they are found in the matrix of mitochondria, and except which one? That is succinate dehydrogenase. Complex one. Now come to this one. This is electron transport system. This is electron transport system located in mitochondrial inner membrane. Electron transport system. Location mitochondrial membrane. Made up of 4 complexes. Complex 1, complex number 2, complex number 3, complex number 4. 4 complexes are there. 2 mobile electron carrier, coenzyme Q, it is also called UQ. Coenzyme Q is also known as UQ, that is ubiquinone. And cytochrome C, this is mobile electron carrier associated with the mitochondrial electron transport system. Mobile electron carrier. First 2 mobile electron carrier, this is also mobile electron carrier. So UQ and cytochrome C, these 2 are the mobile electron carrier. And each complex interact with the proton. Actually, each complex receives electron actually. So see the first complex, complex number 1, it has a enzyme NADH dehydrogenase. NADH dehydrogenase. This is enzyme number 1 of complex number 1. And it has FMN, flavin mononucleotide. And you see this is NADH, this is formed in the Krebs cycle as well as glycolysis both. This H ion, these H ions are released into the intermembrane space. H ions, they are positively charged, whereas electron is negatively charged. Electron is taken up by the FMN, followed by it follows different path and reaches to the complex number 4. Electron released from NADH, that is NADH, if it splits, then what is formed? NAD, then H plus ions. H plus ions are proton. These protons are pushed into the intermembrane space, whereas electron follows this path. This is the pathway of electron transfer. When electron transfer energy is released, that energy is utilized by the pushing these hydrogen ions in intermembrane space. These H ions are released in the intermembrane space. For that they get energy from electron. Then electron reaches to the complex number 4, and complex number 4 comprises of cytochrome A and cytochrome A3. This is the last electron carrier. Whereas complex number 3 has cytochrome B and cytochrome C1. Cytochrome C1. These are the cytochrome associated with the electron transport. NADH when releases their hydrogen or proton plus electron, electrons are passed from complex number 1, complex number 3 and complex number 4. This is the path of transfer of electron released from anions of NADH via UQ 1 to 3, सीधा 1 से 3. Then this is cytochrome C transfers electron from third to fourth, and these electrons are released. Meanwhile, see this one. Protons are collected in intermembrane space. Protons are collected in intermembrane space every time when NADH plus H release their proton. These anions are collected in intermembrane space, and from them these n number of anions are transferred back to the matrix of mitochondria via this one, that is F0 F1. This is F0 F1 part. F0 F1 part, it contains ATP synthetase enzyme. So proton, they are positively charged, they accumulate in intermembrane space, from them they are transferred to the matrix. This movement of proton is known as chemiosmosis. This movement of proton is called chemiosmosis. Movement of proton, that is known as chemiosmosis. This is chemiosmosis. That is movement of proton from pericondyl space or intermembrane space into matrix. Then these protons immediately trapped by hydrogen ions. These protons are immediately trapped by hydrogen ions and combines with the atmospheric oxygen to form water molecule. This water molecule is formed by terminal oxidation. Formation of water molecule is by terminal oxidation. And why protons are transferred from pericondyl space to matrix? That is proton gradient established in between these two. So movement of anions through this structure is facilitated transport also. It's a very good example of facilitated transport also. And this chemiosmotic theory was given by Peter Mitchell. This is electron transport system that is found in mitochondrial membrane. And complex number 2, its direct involvement in the Krebs cycle, that is oxidation of succinic acid into fumaric acid. And the oxidizer in that case is FAD. Succinate dehydrogenase enzyme has an oxidizer, that is FAD. So this FAD is converted into FADH. So this is about the briefing of electron transport system. So come to the first one. Question number 24. This is related with the complex one. Complex one of ETS accept electrons from. Answer, that is NADH2. And this NADH2 is formed in various oxidation steps. This NADH2 is available from various oxidation steps. Of aerobic respiration. That is in glycolysis, and number 2 intermediate step. Number 3, Krebs cycle. In all the cases, that is, if NADH2 is produced, then this NADH2, this is actually mitochondria. This is actually mitochondria, and it has two membranes are there. It has two membranes, and you know the inner membrane has several folds. So every time NADH2 or NADH plus H releases proton in this chamber, that is pericondyal space, whereas electron is transferred to electron transport system. H plus ion and electron. Electrons are carried out by the electron transport system. There is complex one. Its name of enzyme is NADH dehydrogenase and its oxidizer is FMN. Complex one enzyme is NADH dehydrogenase. NADH dehydrogenase, and its prosthetic group is FMN, flavin mononucleotide, receives that is electron from NADH, and hydrogen ions are transferred to the intermembranous space. See the question number 25. Let's see the question number 25. This is complex number 1 and this is complex number 2. In between them, UQ is active. In between them, UQ is active. That is, if electrons are at complex number 1, they are taken up by the UQ. If electrons are there on complex number 2, they are again trapped by UQ. They are again accepted by UQ. Then UQ transfers them to complex number 3, that is first cytochrome B followed by cytochrome C1, B to C1. Then in between them, that is cytochrome C is there. Cytochrome C is there and it transfers them to A, then finally A3. So, if we know the path of transfer of electron, this electron from 1 is received by UQ, UQ transfers it to complex number 3, and from complex number 3 they are taken up by the cytochrome C, and cytochrome C transferred them to A, and A passes them to A3. From A3 these electrons are released, that 2 electrons are released from cytochrome A3 and they combine with the 2 positively charged proton. These protons are transferred from F0 F1 particle. This is F0 F1 particle from which they are removed, and these are the H ions. They are from pericondyal space. So these electrons are finally taken up by the hydrogen ion and oxygen of atmosphere get converted into a molecule of water. So as far as this question, that is reduced coenzyme UQ, that is it has electron, then definitely it passes to cytochrome C. First B1, then C, but cytochrome complex number 3 is not mentioned here. So we can skip that step and directly goes to cytochrome C, and cytochrome C transferred them to complex number 4. This is the answer of this particular question. Terminal electron acceptor. Terminal electron acceptor receives electron from, that is, see this one. This is terminal electron acceptor. This is terminal electron acceptor A3. receives electron from complex number 3, or exactly that is in fourth complex there is a transfer. So finally fourth complex is involved, that is it releases electron to oxygen, releases electron to oxygen. Then during the alcoholic fermentation, now anaerobic activity is there, acetaldehyde is converted into ethanol. See the two-step activity. First of all, glucose is converted into pyruvic acid, and this process is common between aerobic and anaerobic respiration, that is glycolysis. This pyruvic acid with the help of TPP plus Mg, TPP plus Mg converted into acetaldehyde, CH3 CHO, and here what is also used, this is decarboxylation, then this aldehyde is reduced by NADH of glycolysis. This is NADH2 from glycolysis get converted into C2H5OH. So this is the reduction step, and it needs enzyme alcohol dehydrogenase. With this enzyme that converted into alcohol dehydrogenase. Alcohol dehydrogenase enzyme is involved for the reduction of aldehyde into alcohol. Net gain of ATP molecule during aerobic respiration. See this one. Net gain is asked. In glycolysis, 4 ATP are produced, that is gross production, minus 2, that is 2 ATPs are produced by net production, that is NP, net production. And in addition to 2 NADH2 are also formed. This NADH2 enters in mitochondria. But in mitochondria these achenes are taken up by FAD. So it is converted into FADH2. 2 NADH2 converted into FADH2. And each FADH2 gives 2 ATP. So 4 ATPs are formed. 4 ATPs are formed from glycolysis again. So 4 plus 2, that is 6 ATPs in aerobic condition from glycolysis. Then intermediate step takes place in mitochondria. It produces 2 ATP, 2 NADH2. Within mitochondria, it forms 6 ATP. So 6 ATPs from intermediate step. Then 2 Krebs cycles are there. From one Krebs cycle, 12 ATP. From second Krebs cycle, 12 ATP. That is 24 ATPs from Krebs cycle. Then calculate now. 6 plus 6, 12, and 24. Final answer 36. Final answer is 36. That is, keep all the activities in your mind and frame a sheet. That is how many ATPs are contributed by ETS and how many ATPs are contributed by substrate phosphorylation. Then only you are able to say. This is answer given in NCERT also. How many ATPs are produced when one molecule of pyruvic acid? See this one. First of all, this pyruvic acid converted into acetyl coenzyme A. Acetyl coenzyme A, and in that case 1 NADH2 or NADH plus H is generated, and it forms 3 ATP by oxidative phosphorylation. Mitochondria forms bulk of ATP by oxidative phosphorylation by electron transport system. See this one. When these achions are released in mitochondrial matrix via this membrane. While they are transferring, they release energy. That is, energy is released when protons move from pericondylar space to matrix. That energy is utilized in the formation of ATP. So these ATPs are formed here by oxidative phosphorylation process. By which process? They are formed by oxidative phosphorylation. And each NADH2 gives 3 ATP, whereas FeDH2 produces 2 ATP. So come to this one, that is, see here, that is acetyl coenzyme is formed, then acetyl coenzyme oxidizes completely via Krebs cycle, and each Krebs cycle forms 12 ATP, and from intermediate step 3 ATPs are formed. 12 plus 3, that is 15 ATPs are formed from pyruvic acid. Complete oxidation, that means Krebs cycle definitely takes place. So from 1 pyruvic acid 15, from 2 pyruvic acid 30. Energy currency of a cell, that is given for the ATP. This is for ATP, adenosine triphosphate. When this ATP is converted into ADP plus P, in that case 34 kilojoule energy is released. 34 kilo joule energy is released. Mitochondria synthesizes number of ATP, bulk of ATP during respiration, and mostly by oxidative phosphorylation. That's why mitochondria is known as powerhouse of cell, because it generates energy currency ATP and provides them for other vital activity as an ATP source. So these are the energy currency of cell according to Fritz Lippmann. Who said them? That is Fritz Lippmann. According to Fritz Lippmann, these are the energy currency of cell. ATP are known as energy currency of a cell. Now, amount of energy release, I have already given the data, that is 34 kilojoule. Come to the 32nd question. What is the result of respiration? In respiration, materials are consumed. If materials are consumed, then dry weight decreases. Because there is a consumption of organic matter. In respiration. So that's why the dry weight declines in respiration. Anaerobic respiration is also known as, that is intramolecular respiration. I have described with the first question. Intramolecular, that is only one molecule is involved. Then which is the following in listed product fermentation of yeast? Yeast when ferments it forms alcohol. That is the product of anaerobic respiration. And why it forms? That is because of enzyme zymase. This enzyme is responsible for the formation of alcohol. Which one of the following is common intermediate? That is, this is glucose. Glucose form pyruvic acid. Then pyruvic acid oxidation takes place are either aerobically either aerobically or anaerobically. That is in between them, pyruvic acid is the intermediate. So my answer for this question is, that is pyruvic acid is intermediate between glycolysis and CRAP. Then RQ, direct question. RQ, that is the respiratory quotient, volume of carbon dioxide evolved in respiration upon volume of oxygen taken in. This ratio is known as what? RQ. Depends on respiratory substrate and condition of oxidation. For example, question number 37, RQ value in case of aerobic oxidation of malic acid. That is organic acid. Malic acid is organic acid, C4H6O5. If undergoes oxidation, it forms CO2 plus H2O. Forms CO2 plus H2O. That is 4 carbon dioxide is released, then 3 H2O is also released. I mean oxygen is required, that is 3. This is 5 oxygen, this is 6 oxygen, total 11, that is 4, 2 is 8, 3, that is now balance. See this one. Amount of carbon dioxide released 4 and oxygen taken is 3. That's why value of RQ is 1.55, 1.53. That is, we can say 1.33 indicates that is malic acid is organic acid and its value is more than 1 because reason is clear. See the carbon and compare with the oxygen. Organic acids are rich in oxygen. So they need comparatively more energy. For their oxidation. 38th question. A molecule of pyruvic acid subjected to anaerobic oxidation, that means no Krebs cycle, and it forms lactic acid. Up to the see this one. When glucose is converted into pyruvic acid, there are 2 NADH2. But pyruvic acids are 2. And value of ATP is actually for 6 for 2 NADH2. If it enters in mitochondria. But when pyruvic acid subjected to anaerobic oxidation, that is if it forms lactic acid, in that case it uses 1 NAD. It uses 1 NADH2 per pyruvic acid, 1 NADH2. And 1 NADH2, that is 3 ATP will not be produced. 3 ATPs will not be produced. There is a loss of 3 ATP. If it enters in the mitochondria, its value is 3 ATP. But here that NADH2 is utilized in the reduction. So that's why for per pyruvic acid, 1 molecule of pyruvic acid, then there is a loss of 3 ATP molecules. 39th question. Volume of oxygen consumed is more than, that is volume of oxygen respiration is the more than, value of oxygen is more than what? CO2 produced. This is applicable for fats, that is less than 1. It is 0.7 approximate because fats are oxygen poorer, and proteins, they too have less than 1, that is 0.8 or 9, 0.9 approximate. So this question is actually answer should be A and B both. So we can correct it, that is answer is A and B. If fats and proteins are oxidized, in both the cases, amount of carbon dioxide consumed is more than amount of carbon dioxide released. 40th question. RQ for succulent plant at night. Actually, at night, carbohydrates C6H12O6, they oxidize aerobically. They oxidize aerobically to form a malic acid C4H6O5, C6H4SO5 and plus water. CO2 does not release. There is no value of carbon dioxide and oxygen is there. Yes, oxygen is there. We can add it 3 here, 2 here, this is 3 and this is 3. The equation is balanced. So 0 by 3, in that case value is 0. So for 40th question, that is incomplete or partial oxidation of carbohydrate takes place in CAM plant like Opensia, CAM plants like Opensia, then only you see oxygen is taken in but carbon dioxide does not evolve. So RQ value is 0 for this. That's all for the respiration. Thank you.